So hello and good evening, everyone. CCMA Biolog is celebrating its first anniversary this month as a virtual webinar series organized by our students, and we have had an amazing set of thought-provoking and engaging talks the entire year. Thanks and congratulations to the team for the inspiring initiative in this very difficult year, and to the audience and speakers who have given their time and thoughts to the Biolog. So as you all know, our talks are hosted live and also streamed on our YouTube channel. The typical format is about 45 minutes for the talk, followed by a question and answer session. So today we have with us a very special speaker, an evolutionary biologist and geneticist from JNCSR, Bangalore, Professor Amitabh Joshi. Welcome. He's the founding member of the Indian Society of Evolutionary Biologists, fellow of all the three National Science Academies of India, recipient of top awards, and an active academic voice on multiple forums. He currently heads the Evolutionary and Organismal Biology Unit at the JNCSR, and his interests revolve around the interrelations between the genetics of organisms and their ecology and the influence of these factors in their evolutionary path. Today, he will talk to us about some foundational conceptual issues in evolutionary biology and the search for illusory molecular mechanisms for organismal traits. Thank you so much, Professor Joshi, for accepting our invite, and we really look forward to your talk. Please go ahead.
Thank you very much for that kind introduction, Surupi. I just should make a small correction. I am not presently heading the Evolutionary and Organismal Biology Unit. It's my colleague, Professor TNC Vidya, who is the chair of the unit right now. Not that it's important who heads the unit. Okay, so let me just start sharing. So sorry, for some reason it is not showing me the screen that I want to share in the choices.
That's fine.
Yeah, no, it's not even giving me the option to select that screen. It's showing me all other screens. Should we just go ahead with the backup file that you have? It's a little trickier because then I don't have pointer control.
Could you share your entire screen, if not that window, or that's also not coming? Yeah, we're getting something. Yes.
Are you getting the talk now?
Yes.
Oh, wonderful, wonderful. Thank you. So you're seeing the title slide?
Yes, perfect.
Oh, wonderful. So thank you so much once again, and thanks for the opportunity to share some of our thoughts on this very interesting issue in such a wonderful platform. Before I start with the talk per se, I'd just like to point out that although I have the pleasure of delivering this talk here, this is really part of collaborative work that four of us do on many foundational conceptual issues in genetics and evolution under the informal aegis of the Foundations of Genetics and Evolution group. So this is really based on some of the things we've been working on collectively, the four of us. And needless to say, the responsibility for any errors or confusions that arise from today's talk is entirely mine. So this talk is particularly aimed at younger researchers, especially graduate students, and it is sort of an attempt to raise some issues which push you out of your comfort zone of doing biology and actually stepping outside biology and looking in from the outside and trying to critically examine how we do biology and examine it as an outsider would. So in some ways, this is more of a philosophy of biology talk rather than a biology research talk per se. The talk is also imbued with a certain sentiment, which is expressed here beautifully by Mirza Ghalib, who once said, "Kya farz hai कि सबको मिले एक सा जवाब, आओ ना हम भी सैर करें कोहे तूर की." The argument he is making is that, Moses got a certain answer on Mount Sinai, but we may get a different insight. So let's also go and take a stroll on Mount Sinai. And I think the sentiment here is that although we are often tied up with the minutiae of the specific research problems we work on in our own subdisciplines, sometimes it is good to revisit very, very foundational issues that lie at the core of one's discipline. It can be extremely rewarding. So before we go in, let us just talk a little bit about the various senses in which the term mechanism is often used. One is simply in philosophy, a mechanistic philosophy or mechanism as a philosophy is the belief that phenomena can indeed be reduced to simpler material cause and effect chains. And this is not the sense in which we're going to talk about it here, although this philosophical stance imbues many of the things we'll be discussing. Mechanism is also used in a narrower sense as either a descriptive or a specifically material set of cause and effect relationships. These cause and effect relationships are usually relatively simple, relatively linear, and quite often reductionist. An example of this kind of narrow sense mechanism would be a biochemical pathway, for example, linking a mutant allele product to a phenotype. And this is primarily the sense in which I am going to be using the term mechanism for the rest of this talk. Mechanism is also used in a broader sense in many sciences, not just biology, as either an assertive or a conceptual set of cause and effect entailments. For example, one often talks of something like the logistic model of population growth as the mechanism for population dynamics. And the point to note here is that the logistic model is not a mechanistic model, it's a sort of a phenomenological model. It's the component entities that participate in the model as parameters do not correspond to material entities in the real biological world. So I am not going to be talking about mechanism in this broad sense at all. I'm going to be sticking to mechanism in the narrow sense as a material set of relatively simple cause and effect relationships. So what I'm really going to focus on in this talk is the search for molecular mechanisms for organismal phenotypes. I'm not going to, for example, address the search for molecular mechanisms of molecular phenotypes, which is a much less complicated job. And one of the things that triggered this was many of us in conversations with biology colleagues have heard people say something like, I have described this really interesting behavior in response to a certain environment in my model organism, and now I am after the Holy Grail, the mechanism. And this leads one to wonder whether the search for molecular mechanisms of organismal phenotypes is indeed a Holy Grail, which suggests that it's illusory but not particularly harmful and might even lead to some good along the way, although you're not going to find the Grail, because the Grail really doesn't exist. Or alternatively, is it more like the golden deer of our Ramayana, in which case it is an illusion that is positively dangerous because it leads one astray? And so this is going to be sort of at the back of most of what we talk about here, whether this search is like a Holy Grail search or it's like the golden deer in that it is actually misleading and somewhat harmful. So the inducements to give this talk, to sort of paraphrase Aristotle crudely, the proximate inducement is the Holy Grail references that we keep hearing all the time. But there are also two, what you might call ultimate inducements. The first is some aspects of the structure of scientific explanation, and I'll come to that first. So this schematic here is supposed to show the sort of circular structure of how we construct explanations in science. So this red oval on the left is sort of the reality space. This is where real phenomena happen in the real world and we observe them. So our knowledge of what is happening in the real world is really filtered through our sensory apparatus. And then we make a model essentially using inductive reasoning. The model is an entity that exists in concept space in our heads, if you like. And what we try to do is we try to mirror some aspect of the cause effect entailments from the real world in the form of inferential entailments in this concept space. And then we use the model to deduce certain predictions from it. Those predictions become our hypotheses, which we then test either by observation or experiment. And of course, in order to decide whether our observations are in agreement with the hypotheses or not to a reasonable degree, we need to use statistical methods. So one little point I would just like to make here as an aside is that we often hear the term hypothetico-deductive method almost being used as a synonym of the scientific method, and that's not really very accurate, because the hypothetico-deductive method is really one bit in this circle. Model making is essentially also largely induction. So there's a lot more than just deduction and hypotheses and their testing that goes into constructing scientific understanding. So what we have in the reality space is these observed patterns of cause-effect or causal entailments, which can often be quite complex and quite messy. They can involve feedbacks, as you can see in this mess over here. Now, what we do when we try to construct a model is, depending on what question we are specifically interested in and in what context, we focus on some subset of these cause-effect entailments in the real space, and we try to create in our minds a pattern of inferential entailments that in some way is congruent with the causal entailments that we are seeing in the real world. And this is the process of model building, or what people like Robert Rosen have called the modeling relation. Again, I want to make a couple of small points here, especially with students in mind. One is that models are often expressed in mathematical terms, but one should not confuse the model with its mathematical representation. The same model of the same subset of real phenomena can be expressed in different mathematical formulations very often, depending on what it is one is trying to do. And different mathematical formulations can be more or less useful in different contexts. So that is one. The other is that the model need not necessarily be expressed in a mathematical formulation. It could be a verbal model. Indeed, you could have situations where what you are observing in the real world is a set of mathematical relationships and your model is not mathematically formulated. So this idea that there is a reality which is always material and a model which is always mathematical is also a little bit of a simplification and should be avoided. The real point is, where is the reality existing in which space, and where is the model existing which is in concept space. And the fact that your modeling relation is basically trying to establish a congruence between the elements that make up the causal entailments in the real space and the elements that make up the inferential entailment in the concept space. Now, this is important in the context of something we'll get to a little bit later in the talk, but I'll just mention it here and then we'll come back to this point. And that is that if we invoke mechanism as our model, if we invoke mechanism as the rules of congruence between what's in reality space and what's in concept space, then mechanism actually is a very tiny, very specific subset of all possible modeling relationships. And there is a lot of other ways of establishing a modeling relationship which you miss out on completely if you a priori commit yourself to a mechanism. And again, we'll talk about this later. It will hopefully make more sense at that point. The second inducement in the ultimate sense to give this talk is that many of us are worried by what we perceive as an epistemological narrowing of explanation in biology over the past several decades. So a lot of the richness of explanation in biology has collapsed into saying, we found a gene for X. So people find a gene for parental care, people find a gene for risk-taking behavior, etc. And this is what I mean by epistemological narrowing. And you see this in all areas of biology. In neurobiology, you see a narrowing to neurogenetics and to some extent the molecular and cell biology of the neuron. And there's again a little bit of pushback against this now happening. In developmental biology or embryology, as it was more meaningfully once called, after the 1970s, there was a narrowing down to just developmental genetics. And again, for the last 20, 25 years, there has been a pushback against that within the field, pointing out that what happens in the course of embryonic development involves a lot more that we need to explain than what can just be explained by developmental genes. So basically, these are issues that I believe it is important to think about, because in some sense, why we are getting this epistemological narrowing of the richness of potential varieties of biological explanation is because we have committed ourselves wholeheartedly to mechanism as the way in which our modeling relations in biology work. So this is the outline of the talk. I will begin by talking a little bit about the roots of our obsession with mechanism, talk a little bit about illusions of mechanism and how they arise in multiple areas of biology, talk about some really huge elephants in the room that have been around forever, which we have somehow managed to ignore. And these elephants are basically trumpeting out loud to us that there's a problem with mechanisms. And then we'll go into some broader issues, including syntactics versus semantics and contextualizing reductionism versus holism. So the roots of our obsession with mechanism in biology certainly begin with the machine metaphor, or the metaphor of organism as machine, which begins at least as early as with René Descartes in the 17th century. And this is actually from a more contemporary Chinese textbook, just to show that the machine metaphor is alive and well in our pedagogy. And I'm not going to talk too much about the critiques of the machine metaphor, except to note that they began very early and have only strengthened with time. But the point I wish to stress is that the notion that we can meaningfully think about organisms or cells as machines is something we absorb very early in our lives. And because we absorb it very early in our lives, it becomes part of the way we think. It sort of comes naturally to us, and it takes some effort to unlearn it or to critically examine it. And this underlying belief in the machine metaphor explains why you have problems, for example, like very often you hear in talks people starting their talk by saying something like all biology is chemistry, or organisms are basically just molecules plus mechanisms. And these are extremely misleading ways of thinking about organisms. And but as I said, it takes some effort to go beyond this thinking of the machine metaphor. Critiques of the machine metaphor actually came up pretty early, starting with people like David Hume and Immanuel Kant in Germany. And as I said, I won't go into these in detail, but I will just quote from a more recent article summing up this issue. And there have been many articles in the last 10 years on this particular issue. saying this. Overall, it is argued that despite its heuristic value, the machine conception of the organism today is impeding rather than enabling further progress in our comprehension of living systems. And this is the general consensus now among many biologists in many subdisciplines of biology, especially neurobiology, development, ecology and evolution and behavior, and also among historians and philosophers of biology. So let me now talk about what I mean by the illusions of molecular mechanisms for organismal phenotypes. So imagine we have a system here on the top. This is perhaps a cell, perhaps an organism, an individual organism. Of course, in unicellular species, the cell is also the individual organism. So what we have is some input coming in from the biotic or abiotic environment. This red arrow here, there is some input X, and some phenomenon then takes place in the system which gives rise to some output or some observable change in physiology or behavior or whatever that we are referring to as output X prime. But underlying this, what is really happening, if you look at the reality underlying this, you have a complex network of components within this system that have all kinds of cause-effect relationships among themselves. They have many feedbacks. Many of these interactions are highly nonlinear. And of course, you have this very complex network of feedback, nonlinear cause-effect relationships connecting from the red input on the left to the green output X prime on the right. And what we often end up doing when we talk about mechanism is we focus on a subset of these components. In this case, if I just look at the red dot, so from this previous book, I just focus on this red one here, the green one here, the blue one here, the green one there. And I can construct a simple linear chain of cause and effect, which connects me from my input to my green output here. And I have created the illusion of a mechanism. Now, this particular means of creating illusions of mechanisms is very common in biology, not just when one is trying to find molecular mechanisms for organismal phenotypes, but even within organismal and evolutionary biology. So on the left here, you have the canonical textbook depiction of evolution in the lineage of horses, showing what is purported to be an evolutionary trend through time going from the bottom to the top of an increase in size, increase in certain jaw characteristics, a reduction evolutionarily in the number of hooves. And this linear progression from Eohippus to Mesohippus to Merychippus to Pleohippus to modern horses, you can find in most textbooks of biology. And of course, this is also completely misleading. This is an illusion of a linear connection of evolutionary changes. In reality, the evolution of horses or the lineage of horses is much more like a multiple branches of a bushy tree or multiple branches in a bush. And the point here is that many of these branches, if you were to follow the patterns of what's happening to the hooves or what's happening to body size, they go against this simple linear progression. But most of these branches have gone extinct, except for one lineage of modern horses that remained in North America and eventually moved into Eurasia and then even went extinct in North America. And it is only because we are picking up 5 out of these many species and drawing a straight line through them that we are creating this illusion of a linear evolutionary mechanism that was at work, giving rise to consistent increase in body size and reduction in the number of tools. So this kind of illusion is actually very, very common in biology. Now, there are a couple of other things one can do to avoid this problem. So one thing that we often do is, instead of ignoring many of these components and focusing just just on a few of them. We don't ignore the components. We average over all the other components and focused on just these. And then that is how we construct phenomenological or statistical models. And this is quite common in various areas of science, not just in biology. The other thing is that we sometimes get lucky. And the way in which we get lucky is that towards the end of this complex network leading to the output on the right, the green arrow, you have a step, something like saying you take this particular component X prime, and then this violet component here just says multiply the value of X prime by 20, and then you get the output. So if you end up with a simple rule like this, close to your ultimate output that you are measuring, then you have a good chance of isolating this little rule towards the end and indeed finding a simple mechanism that allows you to then target your interventions or perturbations to the simple mechanism and have a very good chance of affecting the output without simultaneously affecting a dozen other things. So sometimes we do get lucky, but most of the time we don't. And the mechanism we look at is largely an illusion. So let me now say something about some of these elephants in the room that we are talking about. First of all is simply the scale of organismal diversity versus the degree of molecular similarity across life forms. That is screaming out to us that explanations for organismal things cannot be coming at the molecular level. There's just too much molecular similarity across very, very diverse life forms. The second example is actually very interesting. We are all familiar with Mendel's 7 trait pairs in pea plants, which were governed by single locus allelic differences. But actually, it turns out that most of these trait pairs were not controlled by a single locus. They behaved as though they were single locus in Mendel's experiment because fortuitously during the inbreeding that Mendel did to produce pure breeding lines, his trait pair lines, for example, the green seed versus the yellow seed line, happened to get fixed for the same allele at all the other loci affecting seed color. And that is why they ended up behaving as though they were single locus differences, but those traits were not actually single locus traits. This was incidentally out in a paper by Raphael Weldon in 1902, which didn't get as much attention as it should have, because this came in the heyday of the biometrician versus Mendelian fight in biology. And the biometricians and Weldon lost that particular fight and the Mendelians won. But Mendel's trait pairs were really not single locus traits. The same kind of complexity you can see T.H. Morgan grappling with in the early writings from the early 1920s, when he is trying to link mutations at a single allele to phenotypic change and realizing that the phenotype is actually the product not just of that one allele, but of multiple alleles at many loci, and there's a lot more intervening. I mean, between genotype and phenotype, not only is there a lot of genotypic complexity involved, but there's also a lot of physiology involved in between and a lot of development involved in between. Most mutations, when they arise, they have huge genome-specific effects, they have huge environment-specific effects, and genome by environment interaction effects on the phenotypes they change. This is now finally being recognized by mainstream geneticists that mutations often have very genome-specific and environment-specific effects. In my own field of evolutionary biology, there is a move going on asking for what is often called an extended evolutionary synthesis, which is underlain by an unease with treating allele frequency change alone as a mechanism for adaptive evolutionary change. In simple laboratory environments, when we do experimental evolution, which is what some of us do, we often find that composite traits like competitive ability in fruit flies or lifespan or rapid development from egg to adulthood. Even in very simple lab environments, very controlled lab environments, you can have multiple phenotypic routes via different sets of traits to evolving increases in composite traits under the same or very similar selection pressure. All of this is telling us that explanation needs to be richer in biology than just molecular or genetic mechanisms. Similar examples are found in developmental biology, particularly the remarkable work by Stuart Newman and Ramrai Bhatt on dynamic patterning modules. And they make the case that if you want to understand what is happening in the development of form in the course of embryonic development, there is a lot more happening at the level of the cells and their interactions with their environment and each other, and a lot of it is mediated by simple physics, not necessarily by the developmental genes. Similarly, there are now in the literature a lot of levels of analysis and biological plausibility debates in neurobiology, which again boil down to the same point. This is something we are all familiar with. Most drugs, especially drugs for diseases that can be treated as or considered to be complex phenotypes, have all kinds of side effects, often very, very unexpected. And the reason why most drugs have side effects is because drugs are typically found based on some illusory molecular mechanism, and then you figure out that you can find some small molecule that impinges on or targets some component of that molecular mechanism. And the problem is it also affects 100 other things. And that is why you have all these peculiar side effects of most drugs, except for drugs where you are lucky enough to be treating something very, very, very simple. Alzheimer's is another very interesting example. There are many, many such examples. You hear all this talk about these amyloid plaques. Many people in reductionist biology spend a lot of time, energy, and money studying them. But it is not even clear yet whether the amyloid plaques are a cause of Alzheimer's phenotypes or the symptom. And in fact, there is good reason suggesting now that they are not actually the cause. Genome-wide association studies, many of you in the audience, I'm sure, know what they are, and you might be aware that there is a lot of literature now on this so-called missing heritability in genome-wide association studies. What does this mean? This means that if you look at heritability as parent-offspring, a measure or a reflection of parent-offspring heritable similarity regardless of mechanism, you get a certain estimate of heritability for a particular composite phenotype. And then if you do the genome-wide association studies and you identify genetic loci connected with it and estimate heritability in the genetic sense from there, it tends to be much, much, much less than the broader estimate of heritability. So there is more to heritability happening than just what can be explained by variation among individual genotypic variants at loci. Personalized medicine today is quite a big thing. A lot of people keep talking about it. And if you think about it, if the mechanism into which we need to intervene in a very specific way with our small specific, is it really a mechanism in the sense that we normally use and understand the work? Similarly, we have what is often called the crisis of reproducibility in biology. There are lots of things written about this right now. And in many respects, I mean, a lot of the problems with reproducibility in biology are because of sloppy or worse, fraudulent practice. I'm not referring to those. But in many areas of biology where the phenotypes are complex, in psychology, for example, in particular, in many aspects of neurobiology, in many aspects of ecology and evolution, often you don't have the kind of reproducibility from study to study which you would expect from an underlying understanding of organisms as mechanisms or machines. And that is because organisms are not machines. Organisms are very actively responding to even small details of how they are handled in the laboratory. A lab population is not just a repository for some proteins and some DNA and mRNA. It is a living thing. It is a holistic thing which has developed in a very integrated way, and very small differences from lab to lab in either the handling of organisms or the genetic makeup of the populations that they are studying, which may in turn have inadvertently resulted from evolving under slightly different handling conditions in previous labs. All of that can give rise to interesting discrepancies in studies of the same question using the same species in different areas of biology. And the root reason why we see this as a problem instead of an expression of that is the way biology is because we think of organisms as machines. And that is why you still see in the titles of papers people saying things like X, Y, Z in the fruit fly or in the mouse or in the worm. There is no such thing as the worm or the fly or the mouse. Each individual can differ in meaningful and often very relevant ways. So let me now turn to syntactics and semantics and say a little bit more about this, because if we go back to what all of these elephants in the room are telling us, what they are in a sense telling us is that there is so much syntactic richness at every level of biological organization, from molecules to macromolecules, organelles, tissues, cells, tissues, organisms, skin groups, populations, communities, ecosystems. There is so much semantic richness in biological systems at every level that it is difficult to capture it through syntactic richness at one or a few lower levels of biological organization. Another way of expressing this dichotomy of syntactics and semantics is to look at this structural hierarchy of biological organization, from molecules to cells to organisms to populations and ecosystems and so on, and realize that if we switch our focus from structure to function, function doesn't show that quite that simple and same hierarchy. Function is actually integrated in complex ways across multiple levels of the structural hierarchy. And that is what we really are coming to with this difference between syntactics and semantics. So these terms first came about in the context of linguistics. So I will start by explaining what they are in that linguistic context. So on the left here, you have 5 sentences in English, Kannada, Persian, Bengali, and Tamil. Phonetically, of course, in terms of the phonology, the sounds that make up these sentences, they are very different. Syntactically, they are also quite different. You have red colored bird in sky is flying. Look at the order of the words, how the verb, the object and subject are by. You have bird of red color. In sky, flight does. It's not flying anymore. It is doing flight. In sky, red bird is flying. Red bird in sky is flying. So the syntax is different. The phonology is very different. But all of them are conveying exactly the same meaning. Their semantic content, the meaning that they are conveying, following the different rules of their respective grammars embedded in the syntax, the semantic content is exactly the same and is also summed up by this non-linguistic picture here. Now, one can have statements that are syntactically correct. For example, the sentence, "the green errors sleep furiously," is syntactically correct according to English syntax, but semantically meaningless. It doesn't convey anything meaningful. If you look at the second sentence here, "the moth got burned in the candle's flame," it is syntactically very similar in English and Urdu, Persian, and Hindi, where in Hindi and Urdu, for example, it would be "Parwana Shama ki law mein jal gaya." Slight syntactic difference, but very similar components making up the sentence, but semantically it maps out to completely different connotations in the two cultures. In English, it's about an insect getting burnt in a candle's flame. In Urdu, Persian, and Hindi, it's about somebody sacrificing everything for love. So the semantic meaning is very different, even though the syntax is somewhat similar. Now, in biology, similar syntax can often point to very different semantics. For example, pleiotropy in genetics or phenotypic plasticity in ecology or functional biology. Different syntaxes can point to very similar semantics also in biology. For example, redundancy, which is very common in biological systems, canalization or phenotypic buffering, very, very common phenomena, where you have different syntactical information all pointing to similar semantics given the context. And sometimes a valid syntax can point to essentially null semantics. For example, null mutations or lethalities arising from null mutations. So why exactly are we getting into this semantics and syntactics point? That is because in hierarchically organized systems like biological systems, so here you have the structural hierarchy of organization of biological systems from molecules all the way to populations and beyond to communities and ecosystems. Basically, in hierarchically organized systems, semantic information at one level often gets incorporated into syntactic information at the next higher level. So if you look at the syntactic and semantic information arising at this level and go to the next higher level or the next higher level, it often gets incorporated into syntactic information at this level. At the same time, semantic information at one level in such hierarchical systems is typically affected by what is happening at multiple higher levels. And in biology in particular, semantics is everything because quite literally context is everything in biology. Anything happening to an entity at one level is hugely affected by what's happening to other entities at that level and at most levels above that. That is the environment in the broadest sense of the word. So again, why is this relevant? This is relevant, and the reason we are framing this in syntactic versus semantic terms is because there are some broader issues of which this is just one aspect. These are broader issues in the philosophy of science: formalism versus phenomenology, mechanism versus vitalism, qualitative versus quantitative understanding, reductionism versus holism, cause versus effect, physics versus biology as exemplars of science, science versus the philosophy of science. And the reason we are focusing on semantics and syntactics is because of the work of Kurt Gödel in the early 20th century, who showed essentially in his famous incompleteness theorems that a consistent system of axioms whose theorems can be listed, etc., there will always be statements that are unprovable within the system. Now, without going into the details of his theorems, let me just say that Gödel's work came in the context of an existential crisis in mathematics due to the discovery of certain inconsistencies in set theory and a couple of other things. And there was a movement in mathematics at the time led by David Hilbert and others towards formalization, and they wanted to make an entirely self-consistent mathematics by formalizing everything. And another way of saying formalizing everything is to say that they wanted to tame semantics and bring it into an expanded syntactic system. And the implication of Gödel's work is that one cannot get rid of semantics in such contexts. And if this doesn't even work for mathematics, it's very unlikely that it's going to work for something as semantically rich as biology. For details of this view of why syntactics doesn't work in biology and why we need to focus on the semantics, I again don't have time to go into the details. I'll refer you to this magnificent book, Life Itself by Robert Rosen, who was somebody who trained both as a mathematician and a biologist and worked most of his life as a theoretical biologist. And he goes into these issues in extremely great depth. I would really recommend this book very strongly to anybody who's interested in these issues, and indeed in the issue of what is life and how is it best studied. So let me end by saying a couple of things about reductionism and holism in hierarchical system. When we talk about holism, what we are saying is that we want to explain things at one level by what is happening at a higher level. That is, we are using the properties of the cell to explain the properties and behaviors of an organelle. That is a holistic approach, jumping over one step in the hierarchy. A reductionist approach would be where we try to look at component parts of the next higher level and explain the behaviors or attributes of the next higher level by the behavior and attributes of components at the previous or greater lower level. That is reductionism. Now, short-range reductionism and holism work reasonably well. If you go up one in your reductionist approach, or you come down one in your holistic approach, go up one from organisms to kin groups, or come down from population. Short-range reductionism and holism work reasonably well. And this point was actually also made in the context of physics in a really interesting paper in 1972 in Science by P.W. Anderson called More is Different. The problem happens when we try to do long-range reductionism and holism, and those don't really work well at all. So if you're trying to do long-range holism, and most people would intuitively see that this doesn't make any sense. If I'm going to try to explain at the level of the organism what is happening at the level of the molecules in its cells, the system is just so rich in higher level layers of semantics, it obfuscates syntactics and semantics at the lower level completely and prevents you from seeing it at all. And most people would agree that this would be really bizarre and silly to try to explain phenomena at the level of molecules within cells by phenomena at the level of the organism. However, the reverse of long-range reductionism is equally bizarre and silly. If you are trying to use phenomena at the level of molecules to explain phenomena at the level of organisms, the explanations at the molecular level are so depauperate in higher level semantics that they will miss everything of importance at the organismal level. So long-range reductionism also doesn't work. So to come back to the original contrast of Holy Grail or Golden Deer, essentially the search for molecular mechanisms for organismal phenotypes or molecular explanations for organismal phenomena is more of a golden deer much of the time, in that it is positively misleading. It leads us astray. It's not just a harmful search for a pleasant illusion. So now the question obviously arises is, if it is so clear that this is a somewhat misplaced search, why is so much effort and money in academic biology invested in searching for molecular mechanisms for organismal phenotypes? And one hypothesis that we would suggest for explaining this is that it is at least in part due to the demands of the pharmaceutical industry. The pharmaceutical industry really is invested in molecular explanations for organismal phenotypes, because if you have a molecular mechanism, then you have a good chance of finding a small molecule which you can use as a targeted intervention or a drug for that particular mechanism, and thereby affect the phenotype in a manner that you want to. However, if you look at the statistics on the pharmaceutical industry, based on data from the 1950s to about 4 years ago, the pharmaceutical industry or the drug discovery industry has by far and spectacularly the worst failure rate of any major or medium industry in the world. 0.14% of all putative drug targets finally make it into the market, and many of them then get withdrawn because of unforeseen side effects. So my hypothesis, or our hypothesis, is actually a good example of how sometimes important and multi-billion-dollar industries can end up shaping not just how we do science, but even how we think about science, and they can have a pretty terrible narrowing effect on how we think about the richness of explanation in biology. So all in all, this pretty much is a picture similar to what is summed up here in these 4 lines by a poet friend of mine, Vibeja Wahid, who says, रेगिस्तां के तपते बालू पे रवाए तिश्न आहू है चमकती रेत जो तुझको नजर आता है आब है जहल तेरा जुनू तू रोक ले अपने कदम वाय नादानी के मंजिल बन चुकी है खुद सुराग. Your goal in itself has become a mirage. And switching to another poet, William Butler Yeats very famously said that poetry is that which gets lost in the translation. And I would like to paraphrase Yeats and say that biology is that which gets lost in the extreme reduction. And this loss of richness has consequences which are summed up in this quote from a 2008 annual reviews of microbiology article by Little et al. The last sentence, they say, the scientific triumphs for many, but the cost was a reduction of emphasis, training, and vision in systems level biology, and with that, a reduced ability to address some of the most important current environmental and health challenges. This sentence rings especially poignantly true in today's situation where we are in the middle of COVID, and in India, we are realizing as a notion that we have a severe paucity of people who understand the semantic richness of epidemiological phenomena. And we are stuck with people who are busy with the syntactics of molecular interventions for viral diseases without any appreciation for the semantics of the complex phenomena that epidemiology involves. Thank you very much for your patience, and I will be very happy to address any questions.
Thank you so much, Professor Joshi, for this wonderful talk. Lots of thoughts going through everyone's mind here. And we can now begin with the question answer sessions. So on the Zoom, we can have questions in the chat or Q&A, and also some questions from YouTube. So let me begin with some of the questions here. And then people can also raise their hands and we can unmute and go ahead and ask your questions.
Sure.
All right, so I'll just take a question from the Q&A first and then we'll open for the Zoom. So insightful talk, how do you understand the quote by Stephen Hawking, a question, what breathes fire into the equations?
I understand it as the semantics. That is what breathes fire into the equation. And I wasn't familiar with this quote, and I'm very gratified that you brought this up, because this mirrors something asked in the Javed Nama by my famous poet, in which he asks, Sher rasoz az kuja yad bagu, as khudiya as khuda yad bagu. And he asks, where does the fire in the poem come from? Does it come from within, from the self, or from outside, from God? So it's very nice to see that there are these parallels across disciplines, if you like. Thank you.
Thank you. So there's another question. Now that we know what the problem is, what would you suggest is the solution, and where do we start? May I request a holistic and an all-encompassing answer? This is from an anonymous attendee.
Well, if we agree as a community that this is a problem, ideally, we need to restructure the way we teach biology right from school onwards. Biology is taught, first of all, as a collection of facts with no attempt to create what you might call a logical structure for the subject. It is not that that logical structure doesn't exist. It does exist, and a lot of it comes from evolutionary insights. But that is not how we present biology in school and in college. So we need a much more balanced way of teaching biology, in which everything from what happens at the molecular level within cells to what happens in a developing embryo to what happens can be treated holistically from the perspective of the living system. For example, one simple point, I have made this in many other talks in a different context. In biology, in some sense, there are only three phenomena in the grander sense of the word. There is development, which takes you from essentially a genome in a zygote to an adult with a phenome, if you like. There is ecology, which maps a fitness function onto the distribution of phenomes in the population. And there is heredity, not necessarily just genetics. Heredity can involve more than just genes. There is cultural inheritance, there is environmental inheritance also. And there is heredity through which the fitnesses of different phenomes, if you like, translate into how they produce a new array of genomes in the zygotes of the next generation. Everything else in biology is a detail. These are the only three phenomena. And this is not to say that the details are not important. All of us spend our life working on one or the other detail in one or two of these three phenomena. And some of those details can literally kill you. But logically, everything else is a detail. The only three phenomena are development, ecology, and heredity, and they are connected through evolution. Because what is happening in the process of evolutionary change over generations is that the ecology is altering development through the medium of heredity. This is the kind of way in which we need to approach biology right from school instead of long lists of terms and facts to make it appear in its own logical coherence and structure. And we certainly need to get out of this bizarre obsession we have in India that molecular biology is equivalent to modern biology. Molecular biology is a very small part of biology. It's a tiny part of biology, and it is even arguable that it's not really biology. Molecules don't really have a biology. They have a chemistry which plays out in the context most of the time of a living cell. They don't have a biology any more than an atom of gold is metallic or is a conductor. Those are properties of the bulk material.
Wonderful. Okay, so we have a hand raised. Mallikarjun, you want to unmute and just ask your question?
Hi, Amitabh.
Hi, Malik.
Wonderful. This was long due lecture.
Thank you.
I would say. My question is that with respect to starting biology education at the school level. So are you suggesting that we should talk more of variability than of typical picture?
Yes, we basically need to reorient everything with variability and hierarchical layers of variability all across the spectrum of structural hierarchy in biological systems. There is variability among adjacent cells in expression levels within the same tissue, for example. There's variability in time, there's variability in space across all levels of biological organization, right from the molecules in the cells to the ecosystems. And we need to build our explanations around a proper appreciation of variability and basic understanding of how the evolutionary process works. Because we need to come to grips with the fact that organisms are not machines that somebody made for a purpose. They arise through an evolutionary process mediated through a developmental process within each generation. And we need to bring all of that together into this consideration. We need to get out of this notion that genes will explain everything, or mRNAs will explain everything, or proteins will explain everything. They won't. They might explain certain things at that level.
Sure enough. I mean, I never believed that genes will explain everything or mRNA will explain everything to begin with. That's why I said this was a long overdue lecture to, especially for younger generation.
If I might just add to that, I am by training a classically trained geneticist with a solid grounding in classical genetics and molecular genetics. And over the years, as I have gone deeper and deeper into biology, I have begun to realize that genetics has begun to exert a very constraining effect on how we think about causation and explanation in biology. I did not start my career being sort of anti-genetics in any sense. I'm a classically trained geneticist.
Oh,
But
no,
I
no,
have now
no.
come to this
And
conclusion.
I know that. Even I am a trained geneticist, but right from beginning, I don't think we ever thought that genetics can explain everything. At least the lab that you headed and I joined as a postdoc. So I don't think we started up with that. I think the problem, as you rightly brought out, was more because of the reductionist view that chemists and physicists and so on that came into biology, trying to pin down a molecule or a gene for the phenotypic expression. That phenotypic variability was lost. Right. So that's the reason I asked. I think we should talk more about the variability rather than about
Oh,
a
absolutely.
specific.
Variability is characteristic of the biological systems at every level.
Absolutely. So we have lost that over the decades, and that's what the
No,
focus.
I agree. Completely.
Right. So thank you. And you brought up that you were trained as a classical geneticist and how your thinking has changed over the years. In fact, the next question sort of asks exactly this. What was the inception point of this idea in your mind? How did this occur in your mind? Is there a single thing that brought this around?
No, there's not a single thing. As I said, this is a bunch of thoughts that have arisen in discussions involving the four of us, myself and my fellow travelers in some of these intellectual journeys, Professor T.N.C. Vidya from GMC, Professor Sutit Dey from IISER Pune, and Professor N.G. Prasad from IISER Mohali, who are part of this FOGEC group. See, we have been in this group re-examining various aspects for the last close to 7 years now. Issues at the core of evolution, genetics, biology. So we have been re-looking at the concepts of Darwinian fitness and whether there are problems with how it is characterized and deployed. We have been looking at giving more emphasis and integrating non-genic forms of inheritance better into, and we are by no means the only people in the world who are thinking on these lines. Many people are. And we have also been thinking in terms of the complexity that gets swept under the carpet in the classical genetics approaches that we read. So when we say, you mutate this allele and you change this phenotype, there is a lot of complexity below that. And for example, if you read T.H. Morgan's writings and how his views on this issue change from the 1920s to the early 1930s, it is fascinating. In fact, this is one thing I would urge younger researchers to do. I appreciate it is hard to do this in the present climate where everybody is banging you on the head to publish high impact papers all the time. That's so sad. But it's very helpful to read the older literature from the 1800s and the early 1900s. So Morgan actually grapples with this complication when he is trying to come up with what we all today recognize as the nomenclature of mutations and the way we name mutations and the symbolism involved and so on. And he tries to initially deal with the mess and then realizes that it is so messy that it's better to sweep it all under the carpet and go with this simple one allelic change, one move in phenotypic space, sort of like a Newtonian analog of one force, and nobody knows what that force is, but the force is defined by the displacement it causes in physical space. And in Mendelian genetics, post Morgan's work, the mutation is defined. In those days, nobody knew what the gene was or what the mechanism of mutation was anyway. The mutation was defined by a displacement in phenotypic space. And it's fascinating to see how Morgan changes his views over a decade on this. And a lot of the variation, the mess, the underlying developmental aspects, the physiological aspects, all get swept under the carpet. And then we end up with the gene for parental care, the gene for heart attack, the gene for gambling, and so on. The gene for lifespan. See, it is very popular in my field. One of the things I worked on in the past is the evolution of aging. Every few years, there's a paper in Nature or Science, somebody has found the gene for lifespan. Used to be earlier in Drosophila, nowadays it's in Cynorhabditis. And after 2 or 3 years, you realize you never hear about that gene again. They just vanish. The reason they vanish in almost all of those cases is that eventually people realize that that gene affects lifespan only in that particular genetic background and only in a particular set of laboratory environmental conditions.
Okay. So actually, I really liked one of the statements I saw in your slides regarding the elephants in the room, that if the mechanism is individual specific, is it really even a mechanism? So what about dynamics in explaining this and touching on an area of my interest? What about epigenetics and regulation? What are your comments on that?
That's an important part of what is happening over and above the genes, but it is again a really tiny part because it's really just changing expression levels of genes. That's still a very tiny part of what is going on even in a single living cell. There's a lot more than just epigenetics. There's a whole physiology happening. And physiology, again, is a very integrative system level thing. Again, I remember having this argument with a biochemist colleague, very distinguished biochemist, when we were designing curricula for ISA Mohali many years ago. And I was saying, you're going to have a 5-year course at the end of which you give a master's degree in biology. How come you have not included a course on physiology? And he says, no, anyway, they have 2 courses in biochemistry. See, this is where the mechanism and the machine metaphor leads us to these kinds of errors. Biochemistry is not physiology. Physiology is a very system level thing. So, I mean, epigenetics is interesting and important. And in fact, now there is evidence that epigenetic marks can actually pass through several meiosis, and therefore there is epigenetic inheritance also. But compared to, for example, if you think about non-genic inheritance, epigenetic inheritance seems to be a very small part of it compared to cultural inheritance and compared to ecological inheritance, which play a huge role in the evolution, especially of behaviors, but even of many physiologies. Ecological inheritance in particular plays a role in the evolution of physiologies as well.
Thank you. Rakhu, you have a question? Rakhu, if you could just...
Hear me?
Yes, can. Go ahead.
Hi, Professor Joshi. I was very fascinating the last 1 hour. I was in a trance.
Thank you.
Now, this is more of a comment than a question. I realize that everything that you've spoken of is something that we should understand better, but we are caught in this vortex of publishing things that we investigate. And if I discovered something and I said, these molecules are involved in this phenomenon, nobody's ever going to publish it. And the journal or the referees are always going to ask for mechanisms. So there has got to be, a way to get around this, that if we are to really put this into practice at the grassroots level, there's got to be a way around it. Otherwise, we're forever stuck in this.
Absolutely. I completely agree. And I'm not suggesting as a pragmatic move, I'm certainly not suggesting that one should change the way one writes one's papers right now. My reason for giving a talk of this kind is to open up maybe a couple of new horizons for the way in which people, especially the younger researchers, think about biology. Changing this mindset is going to take a long time, because this is very deeply rooted in the way we think. And there are multiple reasons for this, which I did not go through for paucity of time. the machine metaphor gets ingrained into us even before our formal education at very, very young ages. That's why it's so hard to give up. See, we can all think of an elderly, perhaps not very educated grandparent or somebody who would, his bones would creak when he got up from the chair and he would say, "Arey, ye machine ab purani ho gayi." It's that deeply ingrained. So changing that is going to take a long time, and hopefully more and more people will write about it, talk about it, and maybe 20, 30 years down the line, we'll actually realize that also this unnecessary focus on molecular mechanisms for organismal things has not really been terribly successful if you objectively look at the evidence. But pragmatically, of course, one should do what it takes to get the papers published. Unfortunately, there is this bizarre pressure, especially on younger people, to publish so-called high-impact things. And I just want to say one thing about this. This bothers me particularly for the simple reason that a young scientist, a PhD student, a postdoc, young faculty, that age cohort in their 20s and early to mid-30s, they are at their intellectual peak for very sound evolutionary reasons. They are at their intellectual and indeed physical peak, and the two are not entirely independent. So these are the people who, if they were to grapple with really deep and tough and fundamental problems in science, they have a much better chance of solving them than people of my age now have. Yet, we are ensuring that these are precisely the young, the most innovative, the most active, energetic intellects have to waste their time pursuing low-hanging fruit that can be quickly done, quickly written up, quickly published because they need to show their 5 papers a year or whatever to their review committee. I think this is both a tragedy and a travesty because we are forcing the best people based on their age, all else being equal, to work on relatively trivial problems. And only people of my age group, when we are sort of comfortable, if I don't publish 5 papers a year also, now nobody is really going to bother about it. But by now, my mind is no longer humming the way it was when I was 26 years old.
Correct.
So this is truly a tragedy, and we have to somehow get out of this.
But I guess in a sense, the two can coexist. So we can continue to do what is required, but stay aware of these, larger issues.
Yes, exactly. That's why I bring them up, so that at least somewhere in the back of one's mind, one should be aware also of some of the limitations of the way in which we are doing our day-to-day science.
Right. And I think that is slowly coming through because over the last several years, we have started to appreciate this concept of heterogeneity. For the longest time, we had this, like you also said, we had this tendency to homogenize everything. Every cell is the same, every tissue is the same, and so on and so forth. And now we know that even in a bacterial culture, every bacterium is different depending on its environment and its physiological state. So
every cancer cell in a tumor is different,
Correct.
which people are again now realizing, and they are building that in and bringing in a lot of insights from evolutionary biology to design more effective therapies for cancer.
So at least in a sense that is coming out. So we are slowly getting out of the stupor and appreciating that everything is not the same. So, thank you for this. And it's going to prompt us to cogitate and chew the cud for a lot of things that we do. Thank you so much.
Thanks a lot. Thanks a lot.
Somdata, you have a question?
Yes, thanks, Ruby. Professor Joshi, what a wonderful discussion we have been having. But I will like to follow up on what just Raghu was picking on. So there are now declarations that internationally many universities are doing. One, for example, is the San Francisco Declaration for Research Assessment, where universities are deciding not to judge scientists and their work based on the impact factor of their publications. Utrecht is also doing something like that. Where do you think India stands at the moment?
See, here there has been some small improvement in this regard that I have personally seen over the last 5, 6, 7 years, but it is in small isolated places. For what it's worth, the science academies of India have sort of issued statements on this matter and talked about the importance of not relying on impact factors and these kinds of bibliometric indices when assessing people for hiring, for promotion, for recognition, etc. And I have had the rather pleasant surprise in the last few years of having sat on some selection committees where committee members were explicitly told that please subjectively evaluate the science and don't go after all this stuff. But I have also been in other committees where we had to fight tooth and nail because the institution wanted to go by impact factors. So in terms of following it up, we are patchy, and still I think many institutions have not internalized this notion. And this is partly, there is a broader problem here. And I have written about this in the past in an article I wrote for the Proceedings of INSA in 2014 called Science and the Sufi Spirit. The broader problem is that the culture of science has been changing over the last 30, 40, 50 years, started in the US, spread to Europe, and spread here. And what is now happening is that we have absorbed many aspects of what one might call the corporate culture, for want of a better word. We are sort of, selecting for, by incentivizing all the wrong things. We talk about productivity of a scientist. Now, productivity at best is a metric for a factory, not for intellectual or creative work. Why the hell are we even using that word in the same phrase as scientist or science? We want, in the name of objectivity, we want metrics by which we can compare and rank scientists and departments and institutions. This is all being borrowed from the corporate culture of the business world. And actually, the beautiful irony is it doesn't particularly work even there. If you talk to senior businessmen, they are horrified at what's happening in the business world as a result of all of this. And it certainly doesn't work in science. But we want this objective metric. And the funny thing is, if you think about it, all these little ironies, yet you set up a selection committee consisting of scientists. For what? If you are just going to rank applicants in order of their H index, a computer program can do that, or in the interests of employment in a country like India with a lot of population, a clerk can do that. Why do you need 7 outstanding scientists of the country spending half a day if all you want them to look at is the H index and you don't want their subjective opinion at all? And the root problem is we cannot assess quality quantitatively through quantitative metrics. We can only assess quality qualitatively, subjectively. Now, the problem with that is that, of course, subjective or qualitative assessments are always open to abuse in terms of nepotism, jealousies, etc., etc. So the question becomes, given that the objective system of quantitative metrics anyway is a disaster, how can we improve our system to at least minimize the possibility of abuse or biases affecting our judgment when we make subjective judgments? And many people in India have thought about this. Several of them have come up with ideas. I remember Professor Gadakkar, when he was president of INSA, saying that what should be done is committees should be large enough to have a reasonable diversity of points of view, and diversity along multiple axes, subdisciplinary axes, nature of institution axes, gender, region, etc. And also the committee's composition should keep turning over rapidly so that the same biases, you kind of average over the biases, if you like. There are some things we can do to improve or to enhance the likelihood that subjective assessment is not getting abused or misused or resulting in biased judgments. But to replace it by this absurd quantitative metric scheme, the minute you have a quantitative metric, it will be gamed. So you have number of publications, and you get predatory journals. I mean, it's completely ridiculous to think that you can quantitatively assess quality. It's just a no-no.
Thank you.
If you have the time, I have a couple of related
questions. Sure.
All right. So Piyush and Anushka, if long-range reductionism and holism doesn't work, then what is the approach to understand biology of life if it's not the mechanistic way? And if all the mechanisms are an illusion, then how do we relate to the reality of these phenomena?
That is a very difficult question to answer even in a few hours. I'll sketch for you the outline of the structure of the kind of answer I believe is relevant to this. And if you are really interested in this, I would strongly urge you to read Robert Rosen's book, What is Life? Basically, the point that Rosen and many others in slightly different contexts are making is that mechanism gives us one way of modeling a living system, which is essentially depauperate. Mechanism is a very syntactic way of trying to draw a congruence between what's happening in the real space and the concept space, those two ovals in that early figure that we had. There are other richer ways of doing relational modeling, which do not involve mechanisms or the machine metaphor. And indeed, Rosen spends much of his book developing those alternate ways. It is not an easy book to read, and there is much in that book that I personally don't understand. Some of the math required goes a little bit beyond my fairly limited mathematical background. But there is enough in terms of take home from that book that even I, as somebody who sort of last studied math in 10th standard, was able to take back from the book, and it has enriched my thinking considerably. So it's about having richer ways other than mechanism of establishing mean causal entailments in the real world and inferential entailments in the concept world.
Okay, thank you. And another question from Rahul Siddharthan. The pharma industry may have a high failure rate, as you mentioned, but there are also many successes, starting from truly single locus diseases like sickle cell anemia. So are there any success stories of the holistic approach to diseases?
Well, the holistic approach hasn't been formally deployed to treat disease in a major way in the last 200 years or so. So we don't know, actually, we don't really have the data. And the people who use a holistic approach, at least in principle, for example, certain traditional forms of wellness and medicine like Ayurveda, philosophically speaking, are taking a holistic approach. So I'm not going into the details of specific practices or specific treatments offered for particular conditions, but their underlying philosophy is fairly holistic. But the problem is that in that form of medicine, there is no concept of rigorously testing how well your interventions have done. It's all anecdotal and often selective. So the tests really haven't been done. We don't have the data for a holistic approach. And certainly there have been many successes wherever there was indeed a simple mechanism into which one could find a simple molecular intervention. There have been successes. I totally agree.
All right. So we can probably go towards winding this up with this question, kind of summing it, that would another way of thinking about this be that studying genetics or molecular phenotypes is merely a first step in understanding phenotypic variation, or would even that be taking it too far?
Well, I'm not sure in what sense the person asking the question is using the term the first step. Is it simply a statement of the sequence in time? Or does first step imply that somehow it is going to be easier or more meaningful to start from here? And I mean, is there a logical reason for you to think that this is the first step, or is it just anything could have been the first step? Because from a logical perspective, I don't think it's a good first step to start with at all. Because biology ultimately is about living organisms. And so we should start with organisms. And organisms don't exist or function in vacuum. So we need to look at functioning organisms in a regular, normal context, an ecological context. They are always embedded with other organisms of the same species, of different species, the environment, the abiotic environment. So I think where to start biology is with the study of organisms. Similarly, genetics, I'll give you a little example if I take 2 minutes more. I have been doing this experiment since 1996 when I joined JNC. So every year we have some new incoming students that have a master's degree in some area of biology or the other. And I asked them at the beginning of my population genetics course, can you tell me what genetics is all about? And the first few years, quite a few students would say it's about heredity and variation. Then by about 2001, 2002, more and more people started saying it's about the genes. Nowadays, at least some proportion of students say it's about DNA. You see the epistemological narrowing happening over the 25 years of my asking this question to incoming students. And the point I try to make to the students is, see, genetics is not about the genes. That's a very weird way to my mind of looking at genetics, because when we take any discipline, any subdiscipline or discipline, we must ask, what are the observables about the real world that this discipline is trying to explain? If you ask that question of genetics, what are the observables that genetics is trying to explain? The observables are that offspring tend to resemble their parents more than they resemble other individuals randomly drawn from the population, and that nevertheless, offspring are not identical to their parents in phenotypes or identical to each other. The minute you realize that this is the fundamental observable about the real world that any science calling itself genetics is trying to address, it's immediately clear that genetics is all about phenotypes and their inheritance, not about genes at all. Now, it turns out that genes do play a major role in how we explain these two observables. But that's not the same thing as saying that genetics is all about genes. Similarly, it is very interesting, if you will allow me to indulge for one more minute. If we ask, what is it that any science calling itself biology must explain or try to explain? There are two kinds of observables here. One set of things is what we might describe under the label of the question, figuring out how living things are living, what makes living things tick. We all know that living things have a whole bunch of characteristics that they exhibit, response to stimuli, reproduction, this, that, whatever. How are living things living? How is this constellation of material things that makes up a cell, how is it actually going about the business of being alive? That is one kind of thing that biology needs to address. The other equally important set of observables that biology needs to address is when we look at the living world, we see three things. One is diversity. The entire world is not just made up of one species of bacteria. There's so much diversity of life forms. We need to explain why, if we are going to call our science biology. Why so much diversity? Second thing we notice is that this diversity is not random. It's grouped together hierarchically in degrees of similarity, which is why you can have classifications, and nowadays you can have phylogenes. There is the possible of this hierarchical organization of life forms with more similar ones closer together in ever more broadly inclusive groups and so on. So we need to explain the diversity of life forms. We need to explain the relatedness of life forms. And any science calling itself biology must also address these things. And thirdly, when we look at any life form, they seem to have traits, whether behavioral, biochemical, physiological, whatever, structural, traits that make them good at living in the particular ecological situation they inhabit. So we need to also explain the adaptedness, by and large, of life forms. Now, this set of questions to explain the diversity, the relatedness, and the adaptedness of life forms, this is what broadly constitutes what you might call organismal biology. And organismal biology itself is a terribly ironic term, and if biologists from 70 years ago came to a department and found that it said evolutionary and organismal biology, they would say, what other biology is there? Biology is about organisms, right? But today, because we have molecular biology, which we have begun to deify, we have to say we are organismal biologists. We are so sorry, but we actually work with real organisms, right? The other side, the questions about what makes a living thing living, how does a living organism tick? Those are broadly being addressed by what you might call cellular biology, molecular biology, reductionist biology, if you want to make a dichotomy. The imbalance that we have today is that instead of recognizing that both of these are equally important parts of what any science calling itself biology has to address, and without appreciating, unfortunately, that the problems that we face as societies, whether it's pandemics or multi-drug resistance or all kinds of societal problems that have a biological angle to them, almost none of them are going to get solved by molecular biology alone. They will require a very organismal, a very ecological, a very evolutionary input as well. And we are realizing that, as I said earlier with COVID today, we hardly have any good epidemiologists in India. We have lots of, but that's not terribly helpful when you are dealing with a pandemic. You need good epidemiologists who understand host pathogen, eco-evolutionary and co-evolutionary dynamics, who understand human ecology in rural versus urban areas, etc. So epidemiology is a lot more than understanding the molecular biology of a virus and understanding the molecular biology of an immune response. So the problem in India is that we are now treating biology as if it only has to address that first set of questions. And we have forgotten about the second set of questions. And we are actually suffering as a society because of this. Multi-drug resistance and its evolution in bacteria almost by definition is an evolutionary biology problem, for example. And again, we are denying ourselves, and the way we teach biology, we are doing a disservice to the younger generation, to our students. We are doing a disservice because around many parts of the world, biology is now becoming much more holistic and much more integrated and giving up some of these mechanistic and reductionist fallacies. We are still strongly into it, especially the powers that be that decide research thrust areas and research funding and curricula for schools and all. They are still totally into these mechanistic fallacies. So we are doing a big disservice to our young biology students by not giving them a proper appreciation of biology as a complete discipline, but just showing them one little part of biology and pretending as though that was the whole of biology. That is the problem with biology in India today. And we seriously need to do something about it because it is damaging our ability as a society to meaningfully deal with many problems that we are facing today.
Thank you. So with all the thoughts that this is going to raise, probably it's okay if students can mail you with some specific questions.
Oh, absolutely, absolutely, absolutely.
Thank you so much. Very kind. So I think with this, we should be ending this session today with deep thanks to you, Professor Joshi, for such an enlightening and I would say thought-provoking talk. And I was really looking forward to this and it's been beyond expectations. A lot of ideas running through.
Thank you so much. Thank you so much. My pleasure. And once again, thanks a lot to the students and organizers of the CCMB Biolog for giving me a chance to talk about some of these issues in such a wonderful platform. Thank you.
Pleasure, sir. Thank you so much. This is a perfect anniversary talk, I feel, for Biolog. My apologies for the oversight earlier in the introduction. Have a great day. Thank you, everyone. See you.
Yeah, you too. Thank you.
Bye.
Take